Hey guys, this is Clayton Fernando. In this video section, I am going to discuss about how the stem cells has a role in providing the immune responses against the infectious diseases. For that, I had chosen the two cases. One is the tuberculosis, that is one of the pandemic and the deadliest re-emerged diseases. So for that, how the hematopoietic stem cell and mesenchymal stem cell in has providing the immune responses and repairs the damaged lungs. We will see. Then another case, that is the recent global pandemic disease, recent breakdown, that is COVID-19. So to understand the cellular mechanism and further research findings, let's have some video clips to understand the stem cells and basics and its types and its function, etc. So I hope you enjoy this section.
Cell is an egg cell from a woman that has been fertilized by a sperm from a man. This single cell, called a zygote, is the first cell in a developing human being. It's also called a totipotent stem cell because it can form any type of cell in the body, as well as the umbilical cord and placenta. After a zygote has divided a few times, it becomes an early-stage embryo called a blastocyst. Embryonic stem cells come from the cells inside the blastocyst. Embryonic stem cells are pluripotent. This means they can form any cell in the body, some organs, such as the skin, after birth and into adulthood. Adult stem cells are multipotent. This means they can only become a few different cell types related to the organ where they're found. For example, the skin contains a small number of adult stem cells that can divide to create new skin adult stem cells. Or they can become more specialized skin cells to replace those that are lost due to cell aging or damage.
Video section we had seen the case of COVID-19, that is SARS-CoV-2. Now we will see move on to the tuberculosis. So how the mesenchymal stem cells, that is MSC, has a role in tuberculosis. So first is what is mesenchymal stem cell? Mesenchymal stem cell are the stem cell which are all not the hematopoietic stem cell, which is present in all the human tissues, which can be easily cultured in large amount in vivo condition. So it is secretes the growth factors, cytokines and chemokines and toll-like receptor, but its mechanisms are still unknown. It has a multipotent function which can be differentiated into monocyte, adipocyte, that is the fat storage, connective tissue, oligocyte and other neuros like that, chondrocyte. Then due to these functions, the mesenchymal stem cell has also been used for treating large number of diseases. Some of the clinical ongoing treatments are cancer therapy, autoimmune diseases, inflammation, transplantation, etc. So this is the markers used for identification of the mesenchymal stem cell. Then the functions of mesenchymal stem cells, it makes the inflammation as reduced form, it modulates the immune cells, and also it fights apoptosis, fibrosis, regenerative diseases in bones and cartilages. It can able to differentiate it, and its self renewal are all the functions of mesenchymal stem cells. So the other functions of mesenchymal stem cells that are that plays in tuberculosis are immunomodulatory, that makes the M1 macrophages, that is classical macrophage, into alternatively activated macrophages. That is so then that it repairs the tissues by cytokines, chemokines, oxidants by release of nitric oxide, tumor growth factor. Then also it replaces the dead and dysfunctional cells or tissues. It also promotes the expansion, and they also inhibit the functions of cytotoxic T cells. So due to this function, and there are researchers suggesting that mesenchymal stem cell has may act as a novel niche for the survival of tuberculosis. So why they are saying means that what the mechanism happens inside is that secreted nitric acid, which suppresses the T cell responses, and also it has low level of MSC2 pathway, and there is none of the pathway function in MSC1. MSC1. So due to this function, it evades the lysosome and antigen present in cells from the tuberculosis. So later that researchers find it out that MSC can act as a double-edged sword to fight against the tuberculosis. How they are saying means the bone from mesenchymal stem cell from bone marrow cannot be targeted by tuberculosis, so it can able to that mesenchymal stem cell we can use it for the repairing the tissues of the TB infected lungs. So that is one of the factor. The another factors are it can be easily cultivated, and it can be easily transplanted either by autologous or heterologous. But they are suggesting that allogenic transplantation is not good, because it makes eventual rejection, and also they tried out that MSC for immunotherapy, but they find out that the reactivation of tuberculosis and the macrophages produced from the mesenchymal stem cell are under the control of the host immune responses, so it can't be able to function effectively. So then the another research that go find it out that toll-like receptor in the mesenchymal stem cell can able to recognize macrophage. So it activates the initiates the phagocytosis and antigen presentation to T cells. That signals will be amplified by DAMP, that is damage associated microbial pattern receptors in the cytosol, which ultimately activates the T cell responses. So the adaptive responses are activated. Then the use of drugs in combination with the things it can able to induce the autophagy. The drugs are rapamycin, metformin, and nitrosamide. So it also induces the autophagy, which makes the MSC to fight and kill mycobacterial tuberculosis. So the BCG, some of the countries, they are immunized as a compulsory by the government. Some of the researchers recently find out also that the BCG vaccinated countries are less infectious, less morbidity and mortality in the case of COVID-19 also. It also provide the immunity not for the specific tuberculosis, it also provide for a large variety of diseases. So the BCG, how it helps in hematopoietic stem cell are that BCG from the that has access towards the bone marrow, which makes the changes in transcription of hematopoietic stem cell and the multipotent progenitor that ultimately produces the cell expansion and enhanced myelinosis. So that and also the BCG educate hematopoietic stem cell by epigenetic modified macrophages, that is transcriptionally the genomically the DNA is not changed, but the phenotype of the macrophages are changed, which makes the better protection against the tuberculosis than the naive, that is wild macrophages. So the cellular and molecular mechanism is in the stem cell, what happens inside is not now still now, but it senses the pathogen present by some of the receptors like toll-like receptor, nod-like receptor, and cytokines, especially in IFN1 and IFN2, and pattern recognition receptor and growth factor, that is granulocyte and granulocyte macrophage called stimulative factor. So in another, they have found out that transcription factors like interferon regulatory factor 8 and CEBP P has a role in initiation of myelinosis, and PATH5 and IRF4 has a role in lymphopoiesis. So we can able to target tuberculosis by this by understanding these things also. The vaccine targeting the macrophages has limited efficiency and limited term of innate immunity memory, so we can able to target by macrophages by the use of long-lived hematopoietic stem cell in bone marrow. We can able to target that diseases are. So, what is myelinosis and lymphopoiesis? Myelinosis is the development of innate immunity cells like neutrophils, basophils, and the eosinophil, like the thing. So the and T cells that is generated from the things are all called the lymphopoiesis. Then let we share some of the results of research findings in the tuberculosis are. mm they had checked the BCG immunization with the intravenous injection and subcutaneous injection, and they showed that this is the diagram they show in the results of the things. So here this diagram represents the intravenous injection is makes the higher access towards the bone marrow. It is the growth of BCG, that is Mycobacterium bovis, is high, but the subcutaneous injected BCG population kinetics is very low when compared to the intravenous injected ones. And also the intravenous injected makes the progenitor hematopoietic stem cell lineage as high when compared to uh, the subcutaneous BCG injected ones in most cases. Then they also compared the uh, hematopoietic progenitor cell quantification frequencies in vitro and in vivo condition. And in vitro condition they analyzed after the fourth hour of immunization, and in vivo condition they checked after the third day. Eh, then they had checked the total number of frequency of progenitor cell production progenitor cell production in intravenous and subcutaneous injected ones in the things the progenitor and the total number of LKS production is high in the intravenous protected one, immunized ones only. So they are suggesting that that intravenous immunized BCG can able has the main access towards the bone marrow that makes the better immune responses against the TB. So if we understand the HSC signature of the changing epigenetic factor and transmissional changes like the things, we can able to target that immunity against the tuberculosis and other main diseases, etc. Okay guys, thank you.